Hello, I'm Carla White and I'll be discussing coronavirus biology. Our learning outcomes today is for students to be able to describe how SARS-CoV-2 gets into the cell, explain how SARS-CoV-2 activates its viral machinery, and students will be able to explain how the infected cell is converted into a virus-making factory. Before we start, I wanted you to share what you know about how a cell can get infected by SARS-CoV-2. You can unmute yourself or type it in the chat, kind of like your base knowledge on how you can potentially be infected by SARS-CoV-2 in the cellular level. I'll give you about 3 minutes. Great! So today we're going to discuss in detail how SARS-CoV-2 get into the cell, which is the name of the virus that causes COVID-19. And then once it is in, how does it activate its viral machinery that is encoded in its set of genetic instructions or genome? And then finally, how does it actually uses its machinery to convert the cell into a virus-making factory? But before we discuss about how SARS-CoV-2 gets into the cell, let's begin first by going over what makes up a coronavirus particle, or maybe we can say that what makes up a virus itself. Well, basically, viruses is something that is made up of genetic instruction or materials, whether DNA or RNA. In this case, our coronavirus is made up of RNA. It is protected by an envelope, and this envelope is a combination of either just protein or proteins and fats. Now, if we slice the virus in half, we can see here this orange strand right here. That's the RNA genome. And it is a large genome, and it is a positive sense RNA, meaning it is actually ready to be read by our protein-making machinery, which is our ribosome. Now, the genome itself is coded in a viral protective protein called nucleocapsid, the one here in blue. And this nucleocapsids helps in protecting the RNA and helps the new viral particles to form after the RNA replication. Now, this is surrounded by a lipid envelope, like I mentioned earlier, and usually this lipid envelope is derived from the host cell itself, because viruses cannot make lipid envelope. Now, two of which are what we call your matrix proteins, and it is shown here in purple. That is a matrix protein, and the one in yellow, that's another envelope protein. And these two helps in the formation of the virus particle and help in RNA budding into them. And then lastly, the third one that we are going to talk about, which we will be focusing on today, would be the spike protein. Now, the spike protein gives the coronavirus its name, or corona means crown, and this is the most prominent feature of the viral particle. And of course, the spike proteins play a critical role for the virus in enabling it to get into the host cell. And it is also a major target of neutralizing antibodies to inactivate the virus. Thus, it is the focus of almost all the vaccine efforts nowadays, because we need to neutralize this spike protein so that it won't get into the cell. So lately there has been a lot of news about antibodies and vaccines. What do you know about the development of vaccines lately? Yes, and it's not really available yet for mass production, but there's efforts in making it into reality pretty soon. So let's take a close look at this spike protein. Now, it is made up of 3 of the same protein. We also call that homotrimers, and it has 2 regions or domains: the upper globular domain and the stalk, or kind of like the lower domain. Now, the upper domain, which is in teal right here, it is kind of like a key looking for a cellular lock, and it only fits with a certain lock, which is called the ACE2 or angiotensin converting enzyme 2. That is the name of the lock, and this is the part of the spike that binds with the receptor. And the lower stalk right here, which is in purple, is what we call your fusion machinery. And this fusion machinery is actually spring loaded. So it's kind of like a jack in the box. And the upper globular part of the protein is actually the lid. And once the lid is off, that's the only time it activates the fusion machinery. So in order for it to be activated, the key must fit into the lock from the host cell with an ACE2 receptor. Now, this protein right here that you see, which is the ACE2 receptor binding protein in the fusion machinery, are playing a critical role in latching into the host cell. So let's summarize what the spike is doing to get the virus into the cell. So the upper globular domain of the spike is the key that locks into the ACE2 receptor. And the only cells that have the receptors allow the virus to interact. Studies show that the ACE2 is actually rich in the upper respiratory tract. And once this happens, it activates the fusion machinery. The upper domain is clipped by a protease enzyme. And the protease enzyme that has been shown to play a major role in clipping off that, activating that spike fusion machinery, is called TMPRSS2. Now, once the head of the ACE is clipped, it allows the fusion machinery to turn on, inserts into the cellular plasma membrane, and brings the viral membrane closer with it and fuses them. This creates a portal between the virus and the cell, so the virus can insert its viral RNA genome into the cytoplasm to start replicating. So now that the RNA is in, now what the virus needs to do is to convert the genome into lots and lots of proteins that are going to do the work to remodeling the host cell and copying the viral genome and assembling the new progeny virions. So how does the virus convert its genome to proteins to eventually replicate the viral genome in assembling new progeny? Well, let's take a look at the genome itself first. Now, this long genome of RNA encodes 13 genes. Now, in this diagram, you see an ORF1A, ORF1B. Now, ORF stands for open reading frame. Open reading frame, the one that is shorter and longer, it actually means that it is ready to be read by the protein machinery or our ribosomes. Now, you have the S genes, the envelope genes, matrix, and so on. So, from 13 genes, at least 27 proteins are made. And how do you make 27 proteins out of 13? Well, the virus do some viral trickery for two reasons. One, it cannot operate the way the cell operates and must abide by it. In our own cell, one gene makes one protein every mRNA. So it is a problem. So the ribosome will find the start codon, starts making the protein, and then stop, and then leave. But for virus, for this coronavirus, it doesn't work that way. And so shown here, which shows the ribosome scans along, then creates the protein. This is a problem for the coronavirus. And since these are series of genes in CoV2, our cells encode one at a time. The ribosomes only see one gene and then leave. So how does the coronavirus go around it? So it uses a strategy where it encodes many different proteins in a single gene. In this giant ORF1A genes, the genes don't have a stop codon. So from gene 1, the ribosome starts encoding, then comes along the next gene, and then the stop codon is not found after gene 1. So up to 11 proteins gets encoded. Same will happen once it shifts to ORF1B. So when the frame shifts, up to 16 proteins are made, and these are all called polyproteins. Now, these proteins are separated by what we call a molecular scissors, and the virus encodes its own protease enzyme that clips along these polyproteins to individual protein to do their own activities. So it is actually a target for antiviral drugs, and this is just one strategy for the RNA or the coronavirus to get 16 or more proteins. Now, what about this right-hand side of the protein, or the genome, rather? Well, the second strategy that that the RNA do is by generating RNA copies they call subgenomic RNA. Now, they are less than full length, and they are nested sets that all have 3' end. Looking at this diagram right here, every gene starts in 5' and ends in 3'. So each gene will keep making the protein from the genes on the right end of the genome. And to do this viral copying, at the end of each boundaries of each genome, each signal called transcriptional regulatory sequence, it gives the RNA copying. It gives RNA copying to stop or go, kind of like a stoplight. But this mechanism allows copying. So after S is being copied, then jumps to the next, then copying again, and so on and so forth. These mechanisms we discuss actually solves the problem of expressing the genes to the right end, and the subgenomic RNA are seen by the ribosome. It'll keep doing that downstream. So, to summarize, in order for your RNA genome to be copied, it is a combination of a polyprotein fusion and subgenomic RNA to be able to generate up to 27 proteins. Now, RNA viruses have their own RNA copying machine, but it makes lots of error, contributing to high mutation rate, but not coronavirus. It has its own proofreading mechanisms. So this might be the reason why COVID-2 has a very low mutation rate, unlike HIV, influenza, or Ebola. Now, while RNA copying machines make lots of mistakes, like I said, it has a proofreading mechanism, and probably that is the reason why the genome of your coronavirus is extra long compared to other coronaviruses. Now, this copy editor actually travels along with the polymerase, and it detects error, like a wrong nucleotide. And this editor will kick that error out and replace it with the correct nucleotide sequence. So now that the coronavirus has made its set of viral machinery, what is it going to do with those machinery? How does it use each of those viral proteins to reorganize the host cell and start making lots of new progeny virions? Well, this remodeling is happening inside the infected cell. It does this by creating rooms where the virus can replicate. These rooms are made from rough endoplasmic reticulum surrounded with ribosomes. It moves and stretches and uses it to form these circular vesicles that form throughout the cytoplasm. These vesicles are called replication and transcription complexes. Now, the reason for making these secret rooms is to avoid the sensors in the cytoplasm looking for foreign bodies that can trigger adaptive immune responses to produce antibodies or killer T cells. When produced, it warns all the neighboring cells to mount their defenses against infection. So aside from the rooms, the virus changed the gene expression of the environment of the host cell. And one way is the expression of this first viral protein called non-structural protein 1, or NSP1, and it restricts cellular gene expression by inducing cleavage of messenger RNA, which is hypothesized to repress translation of mRNA. So the 4TS ribosome, which the major part of it helps in translating messages into proteins and allows the mRNA to feed into the ribosome, But the NSP1, shown here in pink, inserts itself like a cork in reducing the translation of these messages. So the first hypothesis is to reduce that cellular messages, that it could stop the ribosome to prefer to translate into viral protein. The second hypothesis is that this is very good general immune evasion strategy. So the inhibiting host gene expression of the interferon, which is the major SOS signal, can be stopped. So the activation of immune response is stopped. So in short, the infected cell tend to have a pretty limited innate antiviral state due to the NSP1 preventing the translation of the proteins to happen. So the final thing that the virus need to do is to take the copied genome out and assemble its new progeny virions to be released. And somehow they get transported out inside the vesicle covered with nucleoproteins from the Golgi intermediate complexes. Now recall, what is the job of the Golgi apparatus? That's right, it is the FedEx that ships and transports proteins out of the cell. And coronavirus uses the Golgi to ship these progeny of virions out of the cell. And it is a mystery why it does that. Now, these vesicles have been pre-studied with spike proteins, the matrix protein, the envelope protein that is outside of the new coronavirus particle. Now, the nucleoprotein coded genome and the matrix protein drive what you call this budding reaction. If you notice, there's like a cleavage right there that causes the viral genome to be enveloped within these vesicles to form these new particles. Now, budding looks like this. It is pinched off the ERG or Golgi intermediate complexes and would have the series of new virus particles transported through exocytosis-like mechanism to the surface of the cell to be released to go to either infect other neighboring cells or breathe out into the environment to infect a new host. So let's summarize what we covered today from our discussion on the coronavirus biology, which is basically the entire life cycle of the coronavirus inside the cell. We discussed the first thing that happens after a virus is inside the body, where it has to locate the right lock, the ACE2 receptor, thought to be rich in the upper respiratory tract, and that spike protein binds in the lock and key mechanism and break off the top part of the spike, and the fusion machinery latches in the plasma membrane to get into the door. It deposits its RNA genome into the cell using some viral tricks to get 27 or more viral proteins translated from the information, the single incoming RNA, and that is through a combination of giant polyprotein fusion processed and by making subgenomic RNAs. These viral proteins do the work in reorganizing the inside of the cytoplasm of the infected cell and take over by stretching the cellular membrane to make rooms where RNA copying machine will make a copy which has proofreading mechanism to make lots of copies of viral genome and make subgenomic RNAs to make more viral proteins. These proteins are then embedded in new vesicles in the cytoplasm, and the newly copied RNA will use these vesicles as a vehicle, buds into them to form progeny of virions before it is released from the cell. So it actually started in one single RNA and released hundreds of progeny variants from that single infected cell in a remarkably short amount of time, about 8 to 10 hours for the average coronavirus. Now, I'd like you to revisit your response at the beginning of the lesson where you shared how cells gets infected by SARS-CoV-2. How did your knowledge shift after the lesson? You are going to answer this question in our discussion board in our learning management system, and it'll be due in a couple of days. And I also would like to invite you to read this supplemental reading material showing the review of the key differentiating characteristics of SARS-CoV-2. This is from Saranosky, and it's called A Profile of a Killer Virus.